Welcome to Part 3 of Chapter 3, Cell Biology. In this video, we will describe cytoplasm, the composition and functions of the cytoskeleton, define organelle, and describe the functions and structures of cellular organelles. Let's dive in. Cytoplasm is the cellular material outside the nucleus and inside of the membrane. Cytoplasm is composed of cytosol, cytoskeleton, cytoplasmic inclusions, and the organelles. Cytosol is the fluid portion of cytoplasm. Within it are dissolved molecules, ions, and suspended proteins. The cytoskeleton functions to support the cell, keep organelles in place, change the cell's shape, and help the cell to move. The cytoskeleton is made of three main structures. Microtubules are hollow tubes made of the protein tubulin. They create an internal scaffolding, transport substances, and aid in cell division. Microtubules also make up centrioles, spindle fibers, cilia, and flagella. They're shown in the middle of the image. Another component of the cytoskeleton is actin or microfilaments. They provide structure, support microvilli, allow contractility of the structures, and aid in movement. The microfilaments are at the top of the image. Lastly, intermediate filaments give mechanical strength to cells, and they're shown at the bottom of the image. Make sure to take a good look at the image and see how that scaffolding is created within the cell. Let's move on to organelles. Organelles are cellular structures with specific functions. We will now go through each organelle. Make sure to pay attention to the structure and function in the cell. The nucleus is a round organelle typically located in the center of the cell. It functions to control all cellular activities. It has a double-layer membrane called the nuclear envelope, which has openings called nuclear pores. Nuclear pores regulate what can enter and exit the nucleus. The nucleus contains DNA to make specific proteins. There's also RNA inside the nucleus that helps to make the proteins as well. In the center of the nucleus is the nucleolus. This is the location where ribosomes are made. In the image shown, the yellow structure at the end of the arrow is the nucleolus. Surrounding it is the pinkish nucleus. As was mentioned, the nucleus holds DNA. DNA does not just float around as a double-stranded helix. Instead, it wraps itself around histone proteins. At this point, the structure is called chromatin. Chromatin is how DNA is found normally inside of the nucleus. Chromatin can condense itself into very tightly bound structures called chromosomes. You can see these structures in the image. The chromosome is the purple X-shaped structure at the top. During much of the cell cycle, chromosomes are found as chromatin. During cell division, chromatin condenses into the compact chromosomes. Ribosomes are a second organelle. They look like tiny spheres. The red arrows in the image shows these little balls. Ribosomes are where protein synthesis occurs. That's where proteins are made. As mentioned previously, ribosomes are made in the nucleolus. Ribosomes can be free-floating or attached to another organelle called the rough endoplasmic reticulum. The next organelle is the endoplasmic reticulum. These are a network of membranes continuous with the outer layer of the nuclear envelope. The endoplasmic reticulum can be rough, meaning ribosomes are attached, or smooth, meaning it does not have ribosomes. The rough endoplasmic reticulum is where proteins are made, and for this reason, it is found closer to the nucleus. The smooth endoplasmic reticulum functions to make lipids and helps in detoxification within the cell. The smooth endoplasmic reticulum also stores calcium ions within the cell. The next organelle is the Golgi apparatus. It is shown in the images as a green structure. The Golgi apparatus is a series of flattened membranous sacs stacked on each other. Proteins made in the rough endoplasmic reticulum and lipids made in the smooth endoplasmic reticulum go to the Golgi apparatus, where they are modified and packaged for secretion or internal use. Substances are packaged into transport vesicles. Two specific products of the Golgi apparatus are organelles. The first one is called a lysosome. Lysosomes contain enzymes. These digest or break down bacteria, nutrients, and non-functional organelles found in the cell. Another Golgi apparatus product are peroxisomes. Peroxisomes are smaller spheres than lysosomes. Peroxisomes have enzymes that digest fatty acids and amino acids. They are found in locations where the function and detoxification occurs, like liver and kidney cells. The next organelle is a proteasome. Proteasomes are large protein complexes that, unlike lysosomes and peroxisomes, don't have membranes. These barrel-shaped organelles have enzymatic regions on their insides that break down and recycle other proteins. Mitochondria are membrane brown organelles that have very important function of making ATP. ATP is made on the inner membranes of mitochondria called cristae. Cristae have many folds, which increases the surface area available for making ATP. When cellular energy requirements increase, so do the number of mitochondria. Centrosomes are a specialized zone of cytoplasm close to the nucleus where microtubules are formed. In the centrosome are two centrioles arranged perpendicular to each other. Centrioles are cylindrical organelles made of microtubules. They function to influence cell shape and movement. Prior to cell division, centrioles divide and a pair of them moves to the opposite ends of the cell. Here they organize microtubules into a structure called spindle fibers. Some cells have cilia and or flagella. Both of these cellular structures are made of microtubules, are capable of movement, and require ATP for this movement. Cilia are short and numerous. They function to move materials over the cell's surface. For example, cilia are found on the cells lining the respiratory tract, so they can move mucus along the surface. Flagella are longer and look like a tail. There is only one flagella per cell. Flagella are found on sperm, where they function to move the whole sperm cell. Some cells have microvilli. Similar to cilia, microvilli can resemble little hairs when viewed under the microscope. But microvilli are very different. Microvilli are extensions of the plasma membrane, as shown in the image. Microvilli increase the surface area of the plasma membrane, which is important in areas where absorption occurs, like the digestive tract. Microvilli are also much smaller than cilia and do not move. Microvilli are supported by actin protein filaments. Congratulations! We've completed describing cytoplasm, the composition and functions of the cytoskeleton, defining organelles, and describing the anatomy and physiology of these organelles.